Hello, good morning, everyone. Today is a special day. We're going to finish the lectures for the basics of immunology. And starting from the next week, we're going to start the clinical part of immunology. So bear with me, this is the last lecture. And I'll try to put things together during this lecture to make the picture complete for the immune response. So today we're going to talk about the T lymphocytes and T cell receptor, and then we're going to finish with the major histocompatibility complex. Let's talk about the T lymphocytes. So all immune cells are generated initially in the bone marrow, but for the T lymphocytes, after finishing the bone marrow stage, they need to go to the thymus to continue their differentiation. Each cell has a unique T cell receptor. So for the B cell, they have a B cell receptor, which is the antibody. For the T lymphocytes, they have a T cell receptor, and each T cell is responsible for a specific type or recognizing specific type of antigen. The same story for the B cell. The difference between the T cell and the B cell, the T cell cannot recognize any different type of antigens other than peptides. So it recognizes only peptides, and these peptides have to be bound to the MHC molecule. This is in contrast to a B cell. In a B cell, it can recognize a free-floating antigen. It can recognize different types of antigens, whether peptides, polysaccharides, lipids, any type of molecules can be recognized by the B cell. But the T cell, it recognizes only peptides that are bound to MHC molecules. So these are the differences between T and B cell. T cell receptor complex. Let's talk about the T cell receptor, and we call it complex because it consists of several subunits, and also the T cell receptor requires the function of other accessory proteins to conduct the function. The T cell receptor, it consists of two subunits, either alpha or beta, and in this case we call it alpha beta T cell receptor, or gamma delta, and in this case we call it gamma delta T cell receptor. A T cell would present only one type of cells, either alpha beta or gamma delta. One single T cell can have around 30,000 T cell receptors on its surface, and all of these T cell receptors should have either alpha, beta, or gamma delta. The one single T cell receptor cannot mix different types of T cell receptors and cannot mix and match different subunits. So it's either alpha, beta, or gamma delta. So let's compare here, have a slight comparison between the antibody, which is a B cell receptor, and the T cell receptor. You know, roughly they look the same. We can say that T cell receptor can look very similar to this part of a B cell receptor. There is a variable region and a constant region. The same applies here, variable region and the constant region. Here the two subunits formed from a light chain, and this is part of the heavy chain. Here we just have one part. There is no heavy and light. But still we have a variable region and a constant region. This variable region, as you remember, in a B cell receptor, they can recognize the proteins, different types of proteins. For the T cell receptor, again, the variable region is responsible for recognizing both the MHC, because as we said, the T cell receptor recognizes peptide bound to MHC receptor. So a variable region here, part of the variable region is responsible for recognizing the MHC, and we have a region here called the hypervariable region. Now this hypervariable region is responsible for recognizing different types of peptides. This is the alpha beta chains. in a T cell receptor. So this is an alpha beta T cell receptor. And then here it attaches to the cell membrane of a T cell receptor. And this transmembrane region is really short, so it cannot transmit the signal. Once the T cell receptor is bound to the peptide, it cannot transmit the signal inside the cell because this transmembrane region is really short, just 2 maybe amino acids. And this is why it requires different proteins or different molecules to be responsible for transmitting the signal from the T cell receptor here to inside the cell. The same actually applies to the B cell receptor. So this is a B cell receptor, and again you can see the transmembrane region is really short, and this is why a B cell receptor also cannot transmit the signal to inside the cell, and it requires an accessory protein called CD79 that would transmit the signal from the B cell receptor to inside the cell. So they share this property, both the T cell receptor and the B cell receptor. One thing that is specific about the B cell receptor that is not present in a T cell receptor is that a B cell receptor, or let's say a B cell, when it differentiates into a plasma cell, it would be able to produce antibody that has the identical structure of a B cell receptor, but this can be excreted into blood or secreted into the blood. This is not a feature for a T cell. A T cell cannot produce antibodies, and the T cell receptor should always be attached to the T cell. It cannot be released, and the T cell cannot synthesize similar structures to be released into the circulation. So B cell can produce antibodies that are identical in structure to the B cell receptors, but they are in a released form that can be released as antibodies, while the T cell receptors or the T cell cannot produce antibodies similar in structure to T cell receptor. And this indicates the differences in the function between a B cell and a T cell. The T cell has to do the job itself, and the T cell receptor should transmit the signal to the T cell to conduct the function, while the B cell is sort of more resilient. It can act as antigen-presenting cell, and it can act as antibody-producing cell, so it has different form of function compared to the T cell, compared to a T cell. This shows us the gene structure and the protein structure of a T cell. As you can see, it is similar to what we saw earlier in a B cell receptor because they belong to the same family, the immunoglobulin superfamily genes. So they belong to the same family, have similar structures, and the creation of variability in a T cell receptor is somehow very similar to the mix and matching that we saw earlier in a B cell receptor. So this is an example of alpha beta T cell receptor, and we will keep talking about the alpha beta T cell receptor because most of the T cells, about 90% or more of the T cells are presenting alpha beta T cell receptor rather than gamma delta T cells. So this is an example. This is an alpha chain and beta chain, and as we stated earlier, it consists of a variable region and a constant region, and this is a transmembrane region. So this protein is produced from any of the 50 part of the variable region of the alpha chain. We have the junctional area, more than 50 options of the junctional area, and one constant area. So the number of options we have is 50 multiplied by 50 is 2,500 options. For the beta segments, we have the variable region, diversity, junctional, and constant. And you have 50 options of the variable, 2 of the diversity, 13 of the junctional, and 2 of the constant. So by multiplying this, 50 by 2 is 100, 1300 2600 for the beta gene segment, and the multiplication of both options will give us the number of T-cell receptors. This is in addition to the frame shift or the place of the slice where it took place that might produce some extra variation in the T-cell receptor. It is important to note that in B-cell receptor there was another way to create diversity by having a point mutation. So this would increase the spectrum of the possibilities of variable antibodies. This is not present in the T cell, because in a T cell we can look at it as being at a higher level than a B cell, and it is more in a leader position and directing the immune system. It is more in holding the immune response or directing the immune response into a certain direction. So having the chance to develop point mutations is not allowed in a T cell, because the T cell, as we are going to see in the coming few slides, it goes through very rigorous selection process to ensure that this T cell cannot attack self-antigens. So if we open the door for a point mutation, it is possible that after this rigorous selection process, a point mutation might occur, and the T cell then would be able to recognize self-antigen, which will create autoimmunity. So this is another difference between a T cell and a B cell in terms of diversity. So the T cell receptors are not as diverse as B cell receptors. They cannot float in the form of antibodies like a B cell receptors, and they hold higher position. The T cell hold a higher position than a B cell in terms of controlling and directing the immune system. So these are the two segments. These are the genes, the options, the diversity, how the diversity is created in a T cell receptor, but there is no point mutation. So this is regarding the alpha-beta T cell receptor. Let's talk about the different accessory molecules on the T cell surface. Of course, there are accessory molecules, like for example C79, for example on the B cell, but we specifically discuss the accessory molecules on a T cell receptor or on a T cell surface because they play a major role in controlling the immune response, and we will come across these molecules when we are talking about autoimmunity or different types of autoimmune or immune-mediated conditions. You will find that T cell receptor is responsible for many of the immune-mediated or autoimmune diseases, as being the regulator or the major player in the adaptive immune response. And we will come into mentioning these molecules very often. So we have the CD3 complex, CD4, CD8, so we know those. We have CD45, CD28. What is a CD? CD, it's not a compact disk, it's a cluster of differentiation. And cluster of differentiation, it refers to certain molecules. They are cell surface molecules that has been identified excuse me, that has been identified using immunoassays and immune labeling. They managed to identify different types of receptors or molecules on the surface of cells, and they decided to give it names based on the immune staining that they developed. So the CD molecule can be a receptor, can be a ligand, can be an adhesion molecule. So any molecule on the surface of the cell that can be stained with certain immune stains is given the name CD and then given a number. So far, there's around 371 CD molecules that has been identified. So talking about the CD3 complex molecule, as I stated earlier, it's the molecule responsible for transmitting the signal from the alpha-beta T cell into inside the cell. So this is the alpha-beta T cell receptor, and although it shows here that this transmembrane region is a bit long, it's actually really short. It's just 2 amino acid long, and to get the signal deep inside the cell, we need the CD3 complex protein. The CD3 complex consists of 5 different transmembrane proteins. So there's epsilon protein that come in combination with either delta or with gamma. So we have epsilon delta, epsilon gamma, and we have zeta. So it can be 2 zetas or can be zeta and eta. So in total, we have these 5 structures, 4 and 6. So it's either 2 zeta or 1 zeta and 1 eta molecules. Now these molecules, these proteins, once the alpha-beta T cell receptor is activated, conformational changes will occur in the CD3 molecule and would initiate a cascade of events and activation inside the cell. This is for the CD3. CD4, as we know, it's again, it is present on the surface of the T cell membrane, and it is one of the immunoglobulin superfamily genes, so it develops the very similar structures. We can see here the 4 domains, so it is just one single chain with 4 domains, and here the transmembrane region. This is the cell membrane. And the function of a CD4 is, first, it improves the adhesion between the antigen-presenting cell, for example, and the CD4 T cell. So this is the T cell receptor communicating with the MHC molecule holding the peptide. So this is the T cell receptor. Part of it is responsible for recognizing the peptide. Other part is responsible for recognizing the MHC molecule. And this is the CD4 molecule. The CD4 molecule will interact and would recognize the class 2 MHC, would recognize the class 2 MHC molecule, and also it will enhance the adhesion between these 2 cells. And once it recognizes the MHC molecule, it will transmit a signal inside the CD4, and it will activate the CD4 T cell. Also, one particular feature of a CD4 that it acts as a receptor for the HIV virus. So, HIV virus would make communication and contact with the CD4, and then it will attack the CD4 T cell, inserting its viral genome inside a T cell. And this is why there would be a drop in the CD4 T cells in the case of HIV infection, and it is used as a marker to determine the activity of the HIV. We usually do a CD4 count to see if the patient's condition is controlled or if he had developed AIDS when the CD4 count goes below 200 cells. So this is a unique feature for the CD4 T cell. And the reason why they are targeted is the presence of a CD4 molecule that acts as a receptor for the HIV virus. The CD8 consists of 2 chains, alpha and beta, instead of 1, and again belongs to the immunoglobulin superfamily. Only 1/3 of the T cells represents or expresses CD8 on a surface, 2/3 expressing the CD4, and this indicates the difference in the function, as we're going to see later. The CD4 has more variable function and influence on the immune system, and so that we need bigger number of CD4 T cells. Again, function is the same. Here, the CD8, it makes contact with the MHC1 instead of MHC2. It makes contact with MHC1 molecule, and then it will provide the activation of the CD8 T cells, but it does not have the feature of a CD4 of acting like a receptor for the HIV virus. So CD8 is not affected, or CD8 T cells are not affected by the HIV virus, not like CD4. So let's talk about the T cell activation, the antigen processing, and peptide presentation. As I mentioned earlier, T cells do not see the antigen. The antigen, it needs to be a peptide, and it needs to be bound to MHC molecule. T cell only see what is presented by the MHC. If this peptide was floating next to a T cell, it would not be recognized. Processing of the antigen to be presented by the MHC molecule to be seen by the T cell is different between the CD4 and CD8, and this is because the source of antigen is different. So in the CD4 we have, or it follows the exogenous pathway, because the antigen or the protein source is from outside the cell, while the CD8 follows the endogenous pathway because the source of the protein, or the protein, is synthesized inside the cell, as we are going to see here. So this slide here shows us both the exogenous pathway and the endogenous pathway. The exogenous pathway is also known as class 2 MHC pathway because the antigen is presented by MHC2 molecule. So this process happens in an antigen presenting cell, because these cells that are presenting the MHC class 2. So, for example, this is a bacteria, and it was engulfed by an antigen presenting cells. Once it got inside, this is the protein, and it was degraded. It was attached to a lysosome, and these enzymes managed to degrade these proteins. Here are the class 2 MHC molecule is being synthesized in the endoplasmic reticulum. It goes here into the Golgi apparatus to be surrounded by an envelope. This envelope then would become, or this lysosome that contains these proteins is close to the Golgi apparatus, and now this chaperone is removed. We have a free MHC class 2 molecule that will attach to these proteins and present it on the surface. Now we have a peptide with the desired length peptide attached to MHC class 2. Now this is ready for recognition by a CD4 T cell. On the other hand, the endogenous pathway, or we call it MHC class 1 pathway, the virus would be attached to the cell. This is any infected cell. And of course, the virus will get its genome inside the cell, whether DNA virus or, whether a DNA virus or RNA virus. Now this genome will go inside the nucleus, will get itself attached to the cell DNA. It will hijack the ribosomal machinery. It will hijack the transcriptional machinery first to synthesize a messenger RNA. The messenger RNA will be released to the cytoplasm, would be synthesized by the endoplasmic reticulum. The own cells, ribosomes, would synthesize this protein, and now I have a viral protein expressed and produced. this protein will be cut by a machinery known as a proteasome that managed to cut the protein into small pieces. These small pieces now will be attached to the MHC1 molecule and will be presented by MHC1. Now we have the antigen is properly presented using MHC1 molecule. Now this can be recognized by a CD8 T cell, and the CD8 can know now that this cell is virally infected and will come and bind here, get activated, and will act as a cytotoxic T cell and start killing this virally infected T cell. Maybe you've noted this cross-presentation pathway. Remember last lecture we said that a B cell can act as an antigen-presenting cell. So the B cell, or maybe any type of antigen-presenting cell, can engulf this bacteria, present these degrade these proteins. It can present it through MHC class 2, and it also can be presented on the MHC class 1, and in this case the CD8 would be able to recognize the proteins here, which are originated from bacteria. So CD8 T cell can recognize antigens, viral antigens, presented through the endogenous pathway and MHC class 1. Also, CD8, the cytotoxic T cell, can recognize bacterial protein synthesized by the exogenous pathway and presented by MHC class 2, while CD4 T cells recognize only proteins produced by the exogenous pathway that come from the outside and produced or expressed on the MHC class 2. So it is important to recognize that. Okay, in this section, so our lecture is divided into 3 sections. The first section we talked about the B cell receptor. In the second section we're going to talk about the life cycle of the T cell. And the last section we're going to talk about the MHC molecule. So let's talk about the T cell life cycle and development. Remember, this is the bone marrow, the origin of all hematopoietic cells, and the B cell is originating from the bone marrow, goes through certain steps of differentiation, then it moves to complete this differentiation in the lymph node. The T cell also start their life from the bone marrow, start their process of differentiation. It has to go to the T cell, to the thymus, where they go under or go through several steps of differentiation, preparation, and training in the thymus, and then from the thymus it will go to the lymph node to get activated there. So let's see what happens in detail. So in the bone marrow we have the cell is just in a pre-T cell stage. In the pre-T cell there is nothing specifies this T cell as a T cell. It does not have any of the molecules. It did not do any rearrangements in their genes. So it just has the potential to develop into T cell. From that condition, from that stage, it goes to the thymus. In the thymus, it goes through different stages of differentiation and development, starting with the double negative, double positive, single positive, and naive T cell. Let's see what does that mean. And the double negative, we are referring to the CD4, CD8. So there is no CD4, no CD8, and this is why we call it double negative. They have just started arranging the T cell receptor, and it hasn't presented this T cell receptor on the surface. And the double positive, it is presenting both the CD4 and CD8, so this is why we call it a double positive T cell. And now the T cell receptor alpha beta is now fully rearranged and is presented on the surface. A single positive, where it loses expression of one of the CDs, either CD4 or CD8, so it will become only CD4 or a CD8. Naive, it's ready to recognize the antigen, but it hasn't recognized, or it hasn't become in contact with an antigen. Very similar to the virgin type of B cell. And it only gets activated in the lymph node when it comes in contact with the antigen presenting cell. So this is what happens. This is the thymus. Here it's in double negative, still double negative, started presenting the B cell receptor. Here in the double positive, presenting the both alpha and beta T cell receptor, and now in a single positive, so it's either CD4 or CD8, and here it goes into the positive and negative selection, as we're gonna present or show on the next slide. A very important thing to know is that it needs, the first step is to be able to recognize self-antigen. If these cells fail to recognize self-MHC, if they cannot recognize self-MHC, cannot interact at all with self-MHC, so they are useless because they cannot interact with the antigen-presenting cells or with the virally infected cells. They are completely useless, so these cells are deleted. Some of the cells would have very high affinity to self-MHC. They would just bind and stick to self-MHC and would never leave. So these are very risky T cells because they might induce an autoimmune disease because of their high affinity to self-MHC. So again, these are deleted. What we need is a T cell with a moderate affinity to the MHC molecule, so it can recognize the self-MHC, can interact with it, but it does not stick to it. So this is the level of affinity that we need. So let's see what happens in details. Here is the entry of the thymocytes coming from the bone marrow, and as they get here, they reach, this is the thymus here, and this is the cortex, this is the capsule, this is the medulla. So the first step of life, it starts in the cortex, and as I stated, it starts as a double negative. Even the double negative stage, it has different steps, like double negative 1, 2, 3, and it goes all the way, attracted by certain cytokines and chemoattractant to go through this path. Remember that the T cell still is not in a complete T cell form. So at this stage here, when it starts to do the rearrangements of the T cell receptors, okay. So here, as it starts to differentiate further and develop the T cell receptor, it forms the double positive. So now here, still we are in the cortex, the T cell has developed both the CD4 and CD8, so it's double positive, and also developed the T cell receptor. It can be gamma delta T cell, it can be alpha beta T cell, or it can develop into a natural killer cell. We are focusing here on the CD alpha, on the double positive alpha beta T cell receptor. Now this T cell receptor goes into the first step, into the positive selection. A certain types of cell, we call it cortical thymic epithelial cells. These cortical thymic epithelial cells represents a sample of the MHC molecules of the body, self-MHC. And these T cells will come and interact with these MHC1 and MHC2 presented on the cells. So this is the first contact of the T cell with the body, with the cells. If this T cell could not recognize the self-MHC, could not interact with it, it will die, because as we said, they are useless. Only cells that are that have the adequate, moderate binding ability and recognition ability to the self-MHC will be selected. So this is the first step of positive selection. And by this step, we have eliminated between 95 to 99% of the T cells. Only 1% of the produced T cells that come from the bone marrow would manage to go to the medulla. So here we have in the medulla only about 1% of the cells. These cells, but we know that these cells can recognize self-MHC, can interact with the self-MHC, and now they have, decided whether to go into CD4 or CD8. So they are single positive. The next step is the negative selection. And in the negative selection, again, the thymus present a sample of specific types of proteins that the T cell might encounter, specific type of self-protein that a T cell might encounter and should be introduced to. So it will introduce it to insulin, for example. It will introduce it to growth hormone. It will introduce it to liver cells. It will introduce it to different types of proteins, of cells that might be seen by the T cell, so that the T cell recognize these as self. Now, the T cells that have a T cell receptor that can interact with the self protein or self antigens like insulin or Langerhans cells or certain types of antigens or proteins that are presented in our body, in the neuronal system, in the neuronal, in the epithelium, in the cardiovascular, in any part of the body, these proteins should be introduced. So if the T cell has the ability to interact and recognize these self-antigens and interact to it, these would be eliminated. They will not be selected. So only T cells that first were positively selected because they can recognize and interact with self-MHC, and they were selected again because they cannot interact with self-antigens. Now these T cells are ready to migrate and go to the lymph node to meet with antigen-presenting cells. So this is a very important step in T cell development. Actually, it is here where autoimmunity and immune dysregulation takes place. Individuals who have abnormality in the medullary thymic epithelial cells or cortical thymic epithelial cells or genes responsible for presenting different sorts of proteins in the medulla, self-proteins in the medulla to the T cells, if these are defective, the T cells that can come out of the thymus may not be selected rigorously as it should be. They might miss certain self-antigens, and in this case, the negative selection would not be proper and complete. And once they meet these self-antigens in real life, in our body, whether in the pancreas or in the central nervous system, they will start attacking these antigens. So this is very important step in controlling the autoimmunity or autoimmune reaction. The positive selection and the negative selection. Now the cell that got to the lymph node is a naive T cell. They can recognize self-MHC, can interact with it. They do not, we do not have a T cell with a T cell receptor that recognizes self-antigens or self-proteins, so there is no chance for autoimmunity. And they are just ready to face the antigen, the foreign antigen, the bacteria or the viral antigen. And this will be presented to them through antigen-presenting cells, usually dendritic cells in the lymph node. So here we have a naive T cell, and it becomes in contact with antigen-presenting cell that is presenting the peptide through its MHC2 molecule. And now there is an interaction that takes place. This would activate the T cell. The T cell will produce interleukin-2. This is an example of autocrine activity of this cytokine, where a T cell is producing the cytokine to activate itself. Also, the antigen-presenting cells will produce certain cytokines to activate the cell. This cell will proliferate, will differentiate into effector cell or memory cell. And now these activated T cells, for example, here is a CD8 T cell, and here is a CD4 T cell. Now they are activated, ready to go, and they migrate to the site of infection or inflammation, attracted by certain cytokines and chemokines, and it will conduct its function here. The CD8 will recognize the virally infected cell, interact through MHC1, and it will induce killing of that virally infected cell. The CD4 effector T cell will interact with the macrophages present at the site of the infection. It will activate it. It will activate this macrophage. This macrophage will become more aggressive. It will start killing the pathogen. Also, it will activate the macrophage to produce more cytokines that would recruit more inflammatory cells at the site of inflammation. Again, the same picture showing dendritic cells presenting antigens to the naive T cell. And this is the first signal, let's say, to activate the T cell. But we need another signal to complete the activation. Note here the CD28, which is present on the T cell surface, interacting with the B7 on the dendritic cells. This would complete the activation. Now I have an active effector T cell that this T cell would go and interact with a macrophage at the site of infection, or maybe in the lymph node, this active T cell would activate a B cell. Remember that we have the dual relationship between B and T cell in the lymph node. So now I have this effector T cell. It's communicating with the B cell through the CD40 present on the B cell, bind with the CD40 ligand on the T cell. Now this B cell is activated, and I have activated B cell that will go and conduct its function again at the site of infection. So this is how T cell is activated in the lymph node. Maybe you've noted that we mentioned there are more than one stage of activation. The first signal, which is the most important signal, is binding of the T cell receptor with the MHC molecule and transmitting the signal via the CD3 to inside the cell to activate the cell. This actually would not complete the activation of the T cell. This would put the T cell in a steady state, requiring just the last spark to complete the activation. This last spark is the signal 2, or we call the co-stimulation. And the co-stimulation would occur by binding of the CD28, as we saw in the previous image, with CD80 or 86 that is present on the dendritic cells or antigen-presenting cells. So this would complete the activation of the T cell, and now the T cell is completely activated. A CD4 T cell would require another or a further step of activation to allow it to differentiate further or to get or to determine its polarization. Remember that we said that CD4 T cell has the ability to polarize or differentiate into different subcategories. This is determined by a cytokine. So the third signal is the cytokine produced by the antigen-presenting cell. The type of cytokine is determined by the condition of the body, by the type of the pathogen. So this would determine the polarization of the CD4 T cell. Let's see how. So this is a CD4 T cell, and this is the first signal where the T cell receptor recognized the peptide bound on the MHC, and this is the CD4 recognizing the MHC. So this is the first signal. The T cell is in a steady state. We need the second signal, which is the CD28 binding to the CD4 80 or 86. So this is the second signal. Now the T cell is completely activated. If it is a CD8, it would just go straight act or do its function as a cytotoxic T cell. If it is a CD4, it requires the third signal to determine where to go. So the third signal here is the interleukin 12. If the antigen-presenting cell has produced interleukin 12, this would complete the differentiation of the CD4 T cell into a T helper 1. The T helper 1 would produce interleukin 2 and interferon gamma. Interleukin 2 would promote the growth and differentiation of other T cells, and also would promote or enhance the activation of the same T cells by the autocrine effect, remember that we said earlier. And also the interferon gamma will activate macrophages and stimulate B cells to produce antibodies. So this is the T helper, Th1 T helper T cells. The Th2, this is produced, of course, we've got the first signal, the second signal, the third signal is the interleukin 4, which is produced when there is sort of parasitic infection. So when there's a parasite, the cells, the macrophages will produce the interleukin 4. This would stimulate the T cell, the CD4 T cell, to become a Th2 T cell that is able to produce interleukin 4, 5, 13. 4 stimulate B cells to produce IgE, which is important for attacking the parasite. Interleukin 5 activate mast cells and eosinophils, which also are important for parasitic infection. And interleukin 13, which is important for production of mucus in the intestine, which would help to get rid of the parasitic infection. Later, so when we used to study immunology, Th1, Th2 is all what we have. Later, new subclasses and categories of the CD4 T cells have been found. We have the Th17, and the reason why it is called 17 because the T cell would produce interleukin 17. So the trigger for a T cell to become a Th17 is interleukin 6 and transforming growth factor beta. When these cytokines are produced and available, they would push the cell toward Th17 differentiation, and it will produce interleukin 17 that recruits neutrophils to the site of infection, interleukin 22, which will stimulate epithelial cells to produce antimicrobial peptides. The most recent one is the Treg, and Treg because it regulates the inflammatory response. Actually, the inflammatory response needs to be controlled. It's like a beast that you can release to attack a pathogen, but then you cannot keep this beast moving around and damaging everything around it. You need to get it under control. The Treg is the T cell responsible for controlling and regulating the immune response. Once the immune response is completed, like the pathogen has been controlled, different cytokines are being produced, including interleukin-10. Once there is a small amount of interleukin-10, this would tell the T cell that, okay, we are now trying to finish off this job, this inflammatory reaction. So please do not differentiate, do not go into Th1 to produce more interleukin-2, for example. Please do not go into Th2 to produce interleukin-4, 5, and 13. Please do not go into interleukin-17. We want to regulate or reduce the infection. So now it will differentiate into T regulatory, which will produce even more and higher amount of interleukin-10, which is a potent suppressor of the inflammatory response. It suppresses the macrophages, and also it will suppress the transformation of other T cells into Th1 and Th2, Th17. It will recruit more Treg cells to subside and reduce the inflammatory response. So Treg is really important in controlling the immune response. The last type is the follicular type, and this is specifically produced in the germ centers in the germinal centers inside the lymph node, which would guide the B cell to make the appropriate differentiation and antibodies to clear a particular infection. So if there is antigens, certain types of antigens floating, it will help the B cell to differentiate the appropriate B cell of a required antibody to proliferate and to produce antibodies that can bind with these antigens and neutralize them. So this is why we have like 2/3 of the T cells are of the CD4 T cells, because it has different aspects of controlling the inflammatory reaction, either in terms of enhancing the inflammation, or controlling parasitic inflammation, or regulating or suppressing the inflammation, or inducing a humoral type of inflammatory response. So by the end of this section, we've completed the steps that the T cell goes through to become completely activated, to become selected first before activation, to become appropriately selected so that it can conduct its immunological function without affecting the self-antigens or the self-structures, and how it is activated both in exogenous and endogenous pathway, the steps of activation, stage 1, 2, and 3 in the case of the CD4, and the wide spectrum of the CD4 function. The last section, and the shortest, is the major histocompatibility complex. We've mentioned so many of these facts that I'm going to present to you, but just to reinforce the information. So MHC, remember, is the major histocompatibility complex, and when we refer to that, we refer to the molecule or the genes responsible for producing molecules on the surface of the cells in all species. If we are dealing with humans only, as we are going to do for the rest of this course, so we call it HLA. So HLA is the human version of the MHC molecule. As you have seen, MHC is involved, or is the major part for antigen recognition and T cell. Without MHC, a T cell cannot recognize any antigen. And we have different types of HLA. We have and this typing is determined by studying the polymorphisms in the MHC or HLA genes. And studies have shown that certain polymorphisms in the HLA gene are responsible or are associated, not responsible, are associated with certain diseases, with certain autoimmune diseases, or even immune-mediated conditions. So, for example, HLA-DQ8 was found to be associated with type 1 diabetes. HLA-DRB1 1501 was associated with multiple sclerosis. HLA-DRB1 0404 was found to be associated with rheumatoid arthritis. What is the meaning of that number? Numbers and symbols we're gonna see later. The exact mechanism how and why these HLA molecules are associated with particular diseases is not really known. Maybe the shape of the antigen binding area is responsible, but how it does that, how it makes the person more susceptible to develop diabetes, for example, it's not really known. And when we do HLA typing, we are actually trying to figure out what type of polymorphism does this patient have. And this is when we want to do matching of the HLA type. We do the HLA typing to know if for example, this transplanted organ is HLA matched or not. HLA genes are inherited as codominant. You know that you get one chromosome from the father, another from the mother. So you have the two genes, one from the father and the mother. Both of them are expressed equally. And the dominant, we have like this gene is expressed, and this feature is developed because you have this dominant gene. But in the codominance, both are expressed equally. So if you inherited, for example, HLA-A from the mother, HLA-B from the father, you would have HLA-A and HLA-B. Very exactly the same as the blood type, which is again is a codominant inheritance. So if your mother is O, which means there is no antigens, your father is A, B, you will be either A or B. So this is the codominance inheritance. Okay, there are three classes of HLA. There is HLA class 1, which is the MHC1, class 2, and class 3. All of these genes are expressed in chromosome 16, or let's say are located in chromosome 16 in the short arm, and class 2 is the closest one to the centromere. Class 1 is the farthest one from the centromere, and class 3 is in between. So class 1 here is class 1, and we have the classical class 1, which is HLA, B, and C, and we have non-classical HLA, E, F, and G. We have class 2 MHC molecule. Here is the class 2, which is the DPQR. DP, DQ, DR. This is the class 2. Class 3 genes, they are not related to the receptors presented on the cells. It is related to genes encoding for proteins associated with the complement system. So we are not going to talk about class 3. We will talk about class 1 and class 2, which are relevant to our lecture. HLA1 or class 1, they are presented in all nucleated cells, and they consist of alpha chain and just a beta 2 microglobulin molecule, and again similar structure. So it is part of the immunoglobulin superfamily and with the transmembrane domain. And the alpha chain is highly polymorphic. For example, HLA HLA gene, it has more than 200 alleles. So, for example, if I have HLA-A and your typing, your HLA typing is again HLA-A, so we share the same class, but I might be polymorphism 5 and you might be polymorphism 132. So there is 200 different options. This is just for the HLA-A gene. So the HLA class 1 is highly polymorphic. HLA class 2, it is just presented on the antigen-presenting cells, dendritic cells, macrophages, and B lymphocytes. When they act as antigen-presenting cells, they also have, because they act as antigen-presenting cells in some instances, so they also have the HLA class 2 or MHC class 2. And the HLA class 2, it has two chains, alpha and beta. Similar structures, domain with transmembrane area. It is not as polymorphic as the HLA class 1. But the DRB gene is usually polymorphic. There's about 60 alleles in DRB gene. You noticed maybe the numbers that we've mentioned earlier. So, for example, let's see this number here. So HLA, we are referring to the HLA molecule. D is the gene, R is the subregion, because we have DR, DP, DQ. So this is the R subregion. And when we say B, this refers to the beta chain. So this indicates whether the polymorphism is located in the alpha or beta chain. So here it's located in the beta chain in the first domain. And what is the type of polymorphism? It is type 15, variant 1 of type 15. A very close match to it would be HLA-DRB1 1502, for example. So this person is sharing the gene, the subregion, the beta chain, the type, but it's just different in the variant. But it would be just close enough. Of course, there is more detailed classification, but this is what we need to know. For the class 1 HLA, it's a bit easier because we do not have this subregion and we do not have alpha-beta chain. It's only the alpha chain. So when we say HLA-A, so it's the gene A, and it's enough to determine the type and the variant. So this is type 2, variant 1. So this is how, when you see these numbers, you know what it means. It means the HLA typing, and when we do HLA typing, we are trying to get as close as possible to the type of the HLA molecule presented on the cell surface. Remember, HLA class 1 are presented on all nucleated cells, while HLA class 2 are presented only in antigen-presenting cells, macrophages, dendritic cells, and beta cells. So by this, we have completed the basics of immunology. Now we should be able to recognize and know the different types of immune cells, the innate immunity, the adaptive immunity, the cellular part and the humoral part of both the adaptive and the innate immunity. And starting from the next lecture, we will go into the second step, studying basics of immune responses, the autoimmunity and allergic reaction. And the third part of the course would be discussing specific immune-mediated or autoimmune conditions and discuss the clinical picture of these conditions and based on what we know or what we have learned regarding the immune basics or the immunology basics of these conditions. So hope everything is clear to you and you have enjoyed the first part, and hope you will enjoy the second and third part of the course. Thank you very much and see you next week. Bye-bye.